Welcome, everybody. I'm Dr. Mike Tranfalia of FRAXA Research Foundation, and we're here today to talk about clinical trials in Fragile X. And specifically, we want to talk about the clinical trial of Zatolmilast, a phosphodiesterase inhibitor from Tetra Therapeutics. And here with us today are the lead investigator in the trial, Dr. Elizabeth Berry-Kravis. And from Tetra Therapeutics, we have the CEO, Chad Koberly, and we have Francie Kivel, who are going to actually talk to us about some of the specifics of the trial and how you can participate. So with that, I'd like to start with Dr. Liz Berry-Kravis to tell us a little bit about this trial.
Okay, so this is the Fragile X clinical program from Tetra, and I'm going to talk a little bit about how we got to the point of having a Fragile X clinical program at Tetra, and then the earlier phase 2 trial that we ran with this Tetra drug. This goes back quite some time, long ago and not so far away, to Chicago, where I was a fellow doing research on Fragile X syndrome. And I was studying a molecule called cyclic AMP, shown here, which is made in neural cells and acts as a key signaling molecule. And it's made by enzymes called adenylate cyclase, and it's degraded by enzymes called phosphodiesterase. And so the level of this chemical in neural cells is regulated very carefully so that it can kind of control processes that are going on. So we were studying actually another disease, but what we found out when we were studying cyclic AMP in Fragile X is that when we studied patients with Fragile X, they made much less cyclic AMP, shown here in the green line, versus controls, shown here. And this is just the distribution of cyclic AMP. And we didn't quite know what that meant, but we thought it might have to do with cognitive problems in people with Fragile X. So I went to the Child Neurology Society in October of 1990 and presented this finding actually 7 days after my second child was born, shown here. And then we did more experiments. We did a much bigger study and showed that the patients with Fragile X had lower cyclic AMP than people with intellectual disability or autism, as well as controls. And we replicated the study in a much larger population, as well as in multiple tissues, including platelets, lymphoblastoid cells, which are blood cells, and then in fibroblasts or skin cells. And then we thought about whether we could maybe use a phosphodiesterase inhibitor to correct this situation, but there really wasn't one available. So we went on doing experiments, and I went about forming the Fragile X clinic in Chicago based on all the nice families I had met while I was doing these experiments on cyclic AMP. And so all of my work in the Fragile X field can be attributed to these early experiments. But we subsequently showed that if we made neural cells express too much FMRP, which is of course the protein deficient in Fragile X, if there was too much FMRP, there was also too much cyclic AMP. And then Anita Bhattacharya from University of Wisconsin showed that cyclic AMP was low in the Fragile X mouse once the gene had been found and the mouse had been created, and also showed that cyclic AMP was low in stem cells. And then subsequently, Choi et al. Showed that cyclic AMP was decreased in the Fragile X fly. So this is a problem that goes across multiple species. And then it was shown that this could relate, the abnormal cyclic AMP could relate from problems with mGlu-R mediated inhibition of cyclic AMP signaling in the fly. And Jen Darnell showed that FMRP regulates certain adenylate cyclase subunits, as well as probably other proteins in the cyclic AMP cascade. So there's a link between the molecular reason for Fragile X and the cyclic AMP abnormality. So we waited, and every phosphodiesterase inhibitor we reviewed, we looked at several, also had a lot of off-target side effects in the body because phosphodiesterase are important in other tissues as well. And then the wait ended in 2018 when Mark Gurney, who was involved with Tetra Therapeutics, came to visit me in Chicago and said, we have a drug that only inhibits PDE4D, which is a major phosphodiesterase in brain that won't have those off-target effects in other tissues or problem effects in other tissues. And that was BPN 14770. Tetra then conducted studies in the FMR1 knockout mouse, which showed that abnormal cyclic AMP in the knockout mouse was normalized, and that numerous behaviors in the knockout mouse were also normalized, including things like hyperactivity and nesting and marble bearing and social interactions, and also the dendritic spines measured in an additional lab were also normalized on BPN 14770, which is Zatolmilast, the name that Mike tried to say at the beginning, which is now the new drug name for BPN 14770. But I'm going to use this name because it was the name of the drug when we did our trial. So based on all of these positive studies in the mouse, we thought, okay, and also there was an effect in the mouse where if you continued the drug, if you discontinued the drug and then looked at the mouse again 2 weeks later, it had this carryover effect where the drug appeared to work for a longer period of time than it was in the body. So based on the mouse data, we designed a trial, and in designing this trial, we used a lot of the lessons we had learned from the earlier Fragile X trials about 10 years ago, about that we need to understand whether a mechanism that we see in the mouse is going to translate accurately to humans. And so we need to do something that we've kind of, in the field started to call a fast fail trial, where we look at a broad range of different kinds of measures, particularly some measures that don't have a lot of placebo effect, and try to get an idea whether there's any signal with the drug to see if this mechanism is actually going to translate from mouse to man. And we know that we've had experience with the mGlu5 drugs that those didn't translate as well from mouse to man as we would have liked. So we're trying to be careful now. So we designed a crossover study where people were randomized to either placebo or BPN 14770, and they were on this for whatever they were assigned to for 12 weeks. And then they crossed over, and the drug treated patients went to placebo and the placebo treated patients went to drug. And so we wanted to do this crossover study, partly because families always like it when they get a chance to have the drug, but also because we could investigate whether those carryover effects seen in the mouse would be seen in humans. We used the newly validated NIH Toolbox cognitive battery that we had just finished doing a validation study for intellectual disability, and that's an objective cognitive measure that we can use in clinical trials. And then we used the worst problems visual analog scale, where families could rate the problems that they felt were worst for their patients, which is a variation of a measure used in the original Seaside, our baclofen studies. And then we used event-related potentials, or ERP, which is an EEG measure, to look and see if the brain waves are changing when you treat the person with a drug. And we used all these measures to run this intensive phase 2 trial with these objective measures. We enrolled the trial in 1.5 years. We withstood COVID to finish the trial. And yes, we did have to travel to Oklahoma and Miami, right in the middle of COVID when everyone was on lockdown, to get key visits done. But we needed the results from that trial. So we got the results of this trial in early October of 2020. And I know that because the results came out the day before the baby that I took with me to originally present the cyclic AMP results turned 30. So literally 30 years of persistence to get this mechanism tested and get a trial done. And here he is, he's a radiology resident in Wisconsin with his cat. In fact, there was a carryover effect in humans, which I'll show you in a subsequent slide. So all analyses were performed on the first treatment period as pre-specified in the statistical plan. And there were no safety issues and no gastrointestinal problems like usually there are gastrointestinal problems with other phosphodiesterase inhibitors. But this one, because it's specific to brain, did not have those problems in our Fragile X study. And so one of the things that was actually very encouraging and even surprising to us was that all 4 of the performance-based cognitive measures that patients with Fragile X can consistently do on the NIH toolbox, and in particular the crystallized composite, which is kind of a measure of verbal cognition. All 4 of these measures were improved in favor of drug. And in fact, picture vocabulary, reading recognition, and the crystallized composite, which is kind of a combination of those two, showed statistically significant improvement, even just in a 15-patient group of patients treated with drug and placebo. And then we also had this caregiver-rated measure where families identified the problems that their individual had that were most limiting for them. And so we had a daily functions measure, an irritability and anxiety measure, and a language measure. And things, for example, that families flagged as a big problem was getting ready for work, taking the bus on his own, washing his hair or shaving, bathing, showering, doing chores without constant reminders, things like that. And with language, people were rating things like speaking in longer sentences, answering the question, not interrupting, and self-talk, which, as we know, is a problem for many individuals with Fragile X. So they rated what they thought was most problematic for their individual during the study. And actually, the language and daily functioning measures showed significant improvement in favor of drug. Not really significant improvement with anxiety and irritability items that were picked. So this was a reflection of these very specific things that families were rating. And then we did an analysis of Cohen's D, which is a sign of how effective the drug looks like it is. And there were very big effect sizes. These are big effect sizes for the drug on both the toolbox measure and the daily functioning and language measure for the visual analog scale. This is just this just shows the carryover effect. So patients who went on the drug seemed to improve, for instance, on these toolbox tests. And while the patients who weren't on the drug actually worsened a little bit. And I think what we see is that Fragile X patients that feel frustrated with a task tend to not do as well on it the next time because they already know that they don't like it. Whereas we didn't really see that with the treated group. And then the group that was on placebo kind of came up after they switched over to drug. And you can see that pattern in multiple of these measures. And in fact, we saw the same kind of a pattern in things like the daily functioning VAS measure, where the patients on drug went up. And then after switching over to drug, the patients who had been on placebo at the beginning went up. So this is a sign that this looks like there's a carry forward effect. And so we wouldn't be able to use a crossover study design to do a registration trial because there's a very clear carryover effect. But this means that it was the similar finding in mouse and man, which is always nice to see consistencies from one, whenever we see something consistent from mouse to humans with Fragile X, that's a good sign. We had a bunch of other measures that didn't reach statistical significance, but there was a trend in the clinician global improvement measure where we rate all of the various things going on with the patient, where 26.7 improved on drug versus 6.7 on placebo. And that wasn't really statistically significant, but it's a directionality. And then on the KITAP, we actually did see a statistical improvement and this is actually in favor of the drug on the go, no go errors, which is a measure of impulsivity, and maybe some trends on some other things like distractibility. On the ADAMS, the anxiety scale, we didn't see any statistically significant things. And on the aberrant behavior checklist, we saw near statistical significance on parts of the aberrant behavior checklist that measure inappropriate speech and stereotypy, which are not usually the things that we're looking for in trials. Most of the trials we've been trying to improve things like irritability and hyperactivity. So, and this might have been reflected by the seeming improvement in language on the other parent report measure. Now, we didn't see any changes in the Vineland Adaptive Behavior Scale, but the Vineland is a measure that takes a long time to see change, and 3 months is probably not just long enough. On the EEG result, we saw improvement in this right here is the EEG output. And you can see here in the normal situation, the patients and patients on placebo are over responding to sound. This big N1 peak that goes way down here is an over response to sound by people with Fragile X. And on the drug, they had less over responsiveness to sound. So this big N1 amplitude, which is abnormally large in Fragile X, on the drug was almost statistically significant for normalization. And then in eye tracking, we also saw some trends, some movement toward the right direction, with more looking at the eyes in the drug treated group relative to placebo, and less looking at the nose and mouth. And so that is going in the right direction, but it's not a statistically significant improvement. And then some analyses that Lauren Etheridge has been doing on alpha peak, and alpha is a kind of brain rhythm that's linked to cognitive processes, shows some statistically significant differences between drug and baseline in this alpha peak. So these are graphs that just show that. So this was a really unusually positive phase 2 study. We do not have any studies in Fragile X that have had a positive result in a performance-based cognitive measure that's very hard to have placebo on, because most of the patients don't really get placebo on a performance-based measure. And that, if anything, they tend to think, oh, I don't like doing this measure, so it maybe gets a little worse. And on a parent-rated scale of how the patient is doing, and on an EEG measure that obviously isn't going to have significant placebo effects, that looks at how the brain is doing. So this is the most positive phase 2 study we've had in Fragile X. Now, then I have to throw in the caveat that we've had a little bit of trouble in Fragile X, as we all know, moving from phase 2 to phase 3. So we really have to prove that this result that we had in these 30 patients is going to hold up in a large trial. Hopefully we can conduct this trial with a high degree of rigor, like we did the little trial, the small trial at our site, that will be able to demonstrate these effects. We really have to prove these effects are there. With that having been said, we are now conducting this trial, and this is actually the Tetra development plan. And I'm going to turn it over to Chad and Francie from Tetra to talk about the trial and the development program and some of the specifics.
Hello, I'm Francie Kibble, the Executive Director of Clinical Operations for Tetra. And this graphic here represents our phase 2b3 clinical development program. What you see along the top for CNS 204, that is the first of 2 pivotal studies that we're going to hopefully use for registration for this program. The 204 study is in 150 male adolescent subjects, so of the ages 12 to less than 18. We've completed this part at the very top where it says single dose PK. That was an early portion of the study done at Rush and Cincinnati to ensure that the PK profile was what we expected it to be in adolescents, as we'd only previously studied adults. We have now moved into the 3-month treatment period and enrollment phase for 204. Again, this is for males ages 12 to 18, and it will run for 13 weeks. Very similarly, the adult study, CNS 301, is also in 150 subjects. It will continue for the same 3 months, and it's for ages 18 to 45 years. For both studies, we're randomizing in a 2-to-1 fashion, so that 2 out of every 3 subjects that join the study will be randomized to active treatment, and 1 will receive placebo. Upon finishing the 204 and 301 studies at week 13, everyone who completes and remains eligible is open to continue into the CNS 302 open label extension study, right now of 52 weeks in length, where everyone will receive active treatment for the 52 weeks, and we will be doing periodic assessments throughout to look at maintenance effects, as well as for those that receive placebo in the primary studies, how they then do on treatment. Our hope then is to, once we get a positive readout of one of these, the 2 primary studies, is to look at a 3rd study to look at younger children, potentially down to 5 or 6 years old, as well as females of all ages. So that will be triggered based on our outcomes of our first 2 pivotal studies here. And again, those patients would either roll into this same open label study or have the opportunity to do something similar from theirs if we have not continued 302 at that point. That's the overall look at the studies. This is a little more detail about the studies. It's going to share with you a little more about the criteria for eligibility and give you a general feel for who we're looking for. We need patients with Fragile X who have a documented molecular genetic confirmation of FMR1 CGG repetitions of at least 200. They will need to be able to complete the National Institute of Health Cognitive Toolbox picture, vocabulary, and oral reading assessments. You heard Dr. Barry Kravis mention earlier that these were key factors in our phase 2A study that showed very nice statistically significant results. And we wanted to make sure we studied those again and had that opportunity to assess subjects. In terms of other treatments our subjects may be on, they are allowed up to 3 psychotropic medications, and anti-epileptics don't count as one of those psychotropics if they are used for the treatment of seizures. Any psychotropics must be at a stable dose for at least 4 weeks prior to screening and remain stable throughout the study. Anti-epileptic meds, similarly, must be at a stable dose for 12 weeks before screening. Get that longer duration and remain stable throughout the study. Someone who has a history of seizures and is currently receiving treatment needs to be seizure-free for 3 months prior to screening, or if they're not on treatment, they need to be seizure-free for 2 years. Behavioral and non-pharmacological treatments need to be stable for 4 weeks and remain stable. As you can see, we're trying to put a lot of controls around changes in medications over the course of the trial, because that can confound the results. We want this to be as clean and pristine as we can so we get strong results to carry forward. Any subject who is sexually active must be willing to practice barrier methods of contraception. And very importantly, we need to have a consistent caregiver who can attend all the visits. Again, the idea is that the more consistency we show throughout the trial of who's assessing what parameters, the stronger those results will be generally, because variability is smaller. We need that consistency throughout the trial on all kinds of things. We've gotten a lot of questions about where are the sites that these studies are being executed in. This slide shows the currently, I think, 14 sites that are identified. I'd have to count them all up because it seems to change on a weekly basis. But they are identified with the green stars here, and you can see that they track across all of the United States. That was very intentional. We were trying to place sites where everyone would have the opportunity to participate if they wanted to. So we suggest that you look at the map, find the site closest to you, and contact that site if you're interested in participating. We have sites, obviously, at Dr. Barry Kravis's clinic at Rush University in Chicago. We have Cincinnati Children's Hospital with Drs. Pedapati and Erickson at Site 102 in Ohio. We have UC Davis Medical Center out in the more northern part of California with Rondi Hagerman. Children's Hospital of Colorado is number 104 with Dr. Nicole Tartaglia. We have Kennedy Krieger Institute with Dr. Budimirovic on the East Coast, as well as UMass, the medical school, which I know the Tartaglias are very familiar with Dr. Fraser. We have Suburban Research Associates in Pennsylvania, Greenwood Genetic Center with Dr. Buchanan in South Carolina, Emory in Atlanta and in Georgia, and University of Utah, Primary Children's Hospital of Orange County has recently come on board in the Southern California region. And last but not least, we've got the University of Kansas Medical Center with Dr. Genovese at Site 116, smack in the middle of the country. We are hoping to add one more site in Pennsylvania and hopefully another 1 or 2 as we do a little further exploration, but these are the current sites. Now, we often get asked, well, who's open? Well, that changes on a weekly basis, okay? So I would actually recommend you try to find a site closest to you because it'll make it easier for you. There are 6 clinic visits for the pivotal trials where you need to go to the clinic. So it'll be better for your family, I suspect, in most cases, to go to a clinic nearby you. However, if you've been working with one of the sites in the past and you are more comfortable there, that's always something to take into consideration as well. All right, so Chad, I'm going to turn it over to you here to talk a little bit about the potential timelines that we're looking at for bringing this drug to market.
Thanks, Francie. Yeah, so this slide this shows, obviously everyone wants to know what's next after these trials. So obviously we're currently enrolling the trials, as Francie said, as quickly as we can. As we get sites up and running, our goal right now, our target is to complete the trials early to mid next year, 2024, and we would then be applying for what's called an NDA, a new drug application with the FDA, as soon as we can tabulate the data, and assuming it's all positive, probably by the end of 2024, which would then put us into 2025 sometime for approval, in the mid to latter half of 2025, and would launch after that. That's probably the most optimistic timeline at the moment. I know there's questions, obviously lots of people around the world that are interested in this. I can tell you that Tetra is now essentially a wholly owned subsidiary of a company called Shionogi Limited, which is a large Japanese pharmaceutical company that is supporting these trials, as well as very interested in obviously worldwide market potential, in Fragile X for Zatolmilast. And so we are currently starting some of these discussions in some of the other countries. With an orphan disease like this, we're hopeful that most of these other countries are going to accept our U.S. trials, and we won't actually have to run registration trials in all these countries. Proof will find out once we actually have all these discussions with all the different regulatory agencies around the world. But we are starting those discussions now with the EMA, and now that the UK is separate again with the MHRA in the UK. And then we'll be talking to Australia and obviously Japan and other countries around the world. But most of these, hopefully, will be able to use our US data, assuming that it's positive, and we work through some of those issues. We may have to run some small trials in some of these countries, for reimbursement reasons or other reasons. But right now, that's our plan. Our plan is to essentially go global as quickly as we can where there's substantial Fragile X markets. Now, I think we've got some questions.
The first question is, do you think that this drug would improve behaviors and mood and anxiety as well as cognition?
In measures of pure behavior, we did not see improvement in anxiety and mood. I think when we're talking about behavior in Fragile X, though, we're dealing with a rather complex situation, because some of the behaviors are just behaviors that are mediated by FMRP effects, lack of FMRP effects on the brain. But some of the behaviors are either learned behaviors, or they're behaviors that come from lack of ability to, understand, or lack of ability to inhibit a response. And so some of those behaviors may actually be mediated by cognitive processes. So even if behavior is always tied to cognition in these disorders. So it may be that we really just didn't, we were talking about a trial of 15 people on drug and 15 people on placebo. So I wouldn't say it's impossible that behavior could improve, particularly if it's a behavior that's mediated by lack of ability to think through a process, because we do expect that behavior would improve if we improve cognition, but there may be a lag to that while the patient is kind of figuring things out. I wouldn't totally rule out behavior as a target, but I think we don't think of this drug as a primary anxiety drug.
Again, you pretty much answered these, but the questions from people about when other individuals will be eligible for the trials. So when might the trials be opened up to earlier age brackets? You mentioned 6 to 11. What about even earlier? Are there plans for young children?
Sure, I can take that. Yeah, as Francie said, obviously our program right now shows us starting essentially the younger males from 6 to 11, as well as females in that same age range. We are actually actively right now looking at potentially changing the 204 adolescent study to go down to age 9. So it'd be 9 to 18. And we may do that sooner than as shown in the program slide here today. it would probably start early next year while the 204 is still ongoing. As long as they're over a weight range of more than 55 pounds, we're probably going to do that. So that'll be a first little step. Then, yeah, we're waiting essentially for the results of these two trials before we would start the younger kids. There is a chance that we may go younger than 6 as well. Again, I think the issue, and to be honest, right, the lower end is around our endpoints and mainly the toolbox and the ability for the really younger kids to be able to adequately conduct the toolbox that obviously takes some level of maturity to do. So we are going to be relooking at all of that in the next few months as we start to actually design the protocols for essentially what we're calling the 205 study. I know it's a lot of numbers that we're using, but the younger age kid protocol, children protocol in the next few months. So we're going to be relooking at that and challenging ourselves to go as low as we think that we can. And hopefully still have endpoints that are able to be achieved, and maybe use some different endpoints as well as we're starting to discuss this. So unfortunately, yeah, we're going to wait a little bit. Essentially on the timeline, if you think about it, is that we would be starting these studies not too far away, but it'll be about a year and a half. The end of next year, potentially, before we would be starting those younger children studies, assuming that we have positive results in these first two.
Thank you. Then there were quite a few families outside the US, more than I expected to hear from, in all different countries, asking when will these trials be extended to their countries? How does FDA approval apply to other countries if it does? And again, you addressed a lot of this, but it's even beyond the countries mentioned that there are people who are interested.
Yeah, again, I think, as I said earlier, the good thing is that Shionogi is definitely interested in this being a worldwide market. Obviously, Tetra is as well, but Shionogi is supporting that. So we've actually even started some work, because Dr. Barry Kravis herself, a few weeks ago, we had over in London at a symposium where they had doctors from Italy and Spain and across Europe that are learning about this, and pretty much the answer, they were when do we get started, right? Is the feedback that we got from most of them. So I think, yeah, we're seeing the excitement about it worldwide as well. So all I can say is that we will be trying to get into these other countries as quickly as possible. And yes, as soon as assuming that we would get approval in the US, we'll be trying to take that to as many countries as possible that would accept it. And so again, countries like Australia and Brazil and some of these other countries, traditionally potentially can use the all US study results. I know we've gotten some questions too about, starting the trials, these current trials in some of the other countries. And unfortunately, due to our primary endpoint with the NIH toolbox, has not been translated or validated in anything other than sort of US English at the moment. And so we don't want to confound our data in these essentially pivotal phase 3 trials. Obviously, we're in quite extensive discussions even with the US FDA around this endpoint. And so we've decided, unfortunately, to keep the trial just in the US. But I know, I've been in discussion with a few clinicians in Canada, for example, that might be able to refer their patients down to the US to participate, assuming they're good English speakers. But we've decided not to open up any other trial sites, in the UK or Australia, for example, even though they're English. So that's really all I can say, is that we are working on it, and we will be trying to take this, worldwide as fast as we can, hopefully with this US data that we have.
Then another big theme of the questions that we received revolve around the extension trial. So people are worried. They are afraid they're going to have a great response for 12 months, and then it'll be ripped away from them before the drug is on the market. And since there have been some past trials where drugs have worked for some people and they've had to be discontinued. People are really concerned about this and wonder if the 12-month extension could be extended beyond that.
Yeah, that's a good question. And it is something that actually we've been discussing recently as it sort of started coming up from questions from families that are already starting the trial. And so I'm happy to say that we are, obviously it's not completely approved yet, but we are working to extend. We will probably do an extension of the 302 study for at least 2 years, which would make it essentially a 3-year study at the moment, that hopefully would get us through close to approval. And if we don't, then we'll probably, I'm sure, look at extending it again. Because we do appreciate that once families and patients go on drug, they want to stay on it if it's working. And so we obviously hope that's the case as well. And we will commit to probably to support those open label studies at least through approval. But right now we're working on a 2-year extension, which would make it a 3-year study, which would take us into well into the end of 2025, essentially.
I think that's going to be a big incentive for people who are scared.
Yeah, that's understandable.
There seems to be a fair amount of fear, and that's because of past trials that you had nothing to do with, but it happens. So that'll be helpful. Great. And I know that requires some approvals, but if and when you do get that, that's something we should announce as, great news. Look what
Yeah, for sure. We're literally working on the amendment to the protocol right now.
Right.
And Francie can tell some more about we're trying to change some of the endpoints and things in that trial too, to make it a little easier for patients in the extension, which will help also. And then once we get the protocol done, then obviously it has to go through the sites and approved and all that. But our goal is obviously, since we have patients that actually started enrolling in the trials at the end of last year, right? Essentially their first 12-year period will be up in January of 2024. So our goal obviously is to have the extension in place by January of 2024. But the full details we'll probably have out, later this summer as to that, yes, we're moving forward with the extension, and here's what all the new endpoints look like.
Thank you. Other questions were about the requirements for the study, and it sounds like you may be addressing those now. Yeah, go for it.
We have been getting feedback from both our sites and what they're hearing from their families, that the number of visits are a little heavy. Okay, it's hard to get back if you're traveling some distance. So in particular for the open label extension, right now the first 12 weeks mimic the pivotal trials with the visits and the schedule and everything, because some of those patients have never been on drug before. So they've been from placebo. So we wanted to give them the same safety follow-up as the patients in the pivotal trials. But after that, they're still being seen every 4 to 6 weeks. And that may be a bit much. So we're actively working with our medical colleagues, both in Tetra and in Shionogi, to look at what can be done. Can we do some of these virtually? Are they all necessary? Could a visiting nurse do some of this? So we're actively looking to reduce the number of visits that have to be done on site. I know that once every quarter, we would like to have the cognitive assessments done. So, but if we can get it to be one visit every quarter instead of every 4 to 6 weeks, that's certainly an improvement over where we were before. So more to come on that. As Chad noted, we will know exactly where we're headed with this road a little bit further down in the summer. But at the end of the summer, we should be able to give you a very good picture of what we hope to implement there.
I have a question that parents are always asking us, and that is specifically, what are the subjects in the trial required to do? And we're not talking about inclusion criteria, and we're not talking about the assessments that they have to do. We know they have to do the NIH toolbox.
Okay.
What do they have to do as far as taking pills, getting blood drawn, EKGs? What do they actually have to do when they come into the clinic? What is going to be done to them?
Yeah.
I guess is another
Oh
question.
boy, oh boy, that's a big question. I will hit it at what I think is a reasonable level. How's that? And
Okay.
Dr. Barry-Kravis, if you want to chime in too, that's great.
Okay.
All of our treatments are capsules. Okay. And so for the adults, they're required to be able to swallow the capsules. That's part of the eligibility criteria. So that'll be their medication. They'll take it in the morning and they'll take it in the evening before they go to bed. More specific instructions will be provided, but essentially that's it. We really want our adolescent subjects to do the same thing, to take the pill. Okay, take the capsule. It's a small one. It's not one of the big horse pills. It's little. But again, that's how we intend to have it administered. There's a little bit of flexibility if there's an adolescent that just absolutely cannot swallow a pill. We've asked the investigators to talk to us, and we have the option, if necessary, to open the capsule and sprinkle it on some applesauce and administer it that way. So in terms of the drug administration, that's it. The younger people in 204 will titrate up. They'll take 1 pill a day for 7 days, and then they will go to the 2-pill regimen that everybody else will be on. Okay, and if their investigator feels it's necessary due to side effects, which we haven't seen at all in the patients we've treated so far, we've seen very little happening in the terms of side effects. You can step back. We can step back to once-a-day dosing if the PI feels it's necessary. So that's the dosing side of it. There are blood draws at every clinic visit, okay? That's for us to be able to, per the FDA, keep an eye on the liver transaminase functions and all of the other safety functions in the body. So those are necessary. ECGs don't happen nearly as frequently, and those aren't so bad because they're just little stickies. But there's a couple of them done over the course of the studies. Dr. Barry-Kravis, do you want to add anything to that?
No, I think that's pretty much it. That the patient has to come into clinic and they have to be willing to be, to have a physical exam. And the blood test is probably the biggest issue. And we know from our experience with Fragile X that there are certain people that really are not bothered at all by blood tests, and especially the adults have sometimes adapted to it, and they come in and there's certain adults that have been to see me for enough trials that they actually think they're going to have a blood test when they come in, and they're kind of unhappy if they don't get to have a blood test. But but those are the fewer and not the more frequent situation. Some of the adolescents are a little bit harder to get the blood from. I mean, we have various processes. Some would rather go to the blood lab. Some want me to draw it personally. Some like to use cream on their arm, and we are allowed to do that. We can give if it turns out that the lidocaine cream is helpful, we call it the magic cream. If it turns out the magic cream is helpful, then we actually give the family the cream, and they can put it on before they come if that's going to be helpful. So there's a variety of ways of handling it, but in the end, push come to shove, we have to get the blood tests. Usually the EKGs aren't as much of a problem, but there's some patients that are very sensitive to the stickies and don't like them. So that they have to get through. And then they have to be willing to sit down and take the cognitive test. And we have some patients that are really good with all the medical procedures, but they don't want demands put on them. So they don't want to do the NIH toolbox because they're going to have to do work to do that. It is on an iPad, and it's the kind of, it's not as test, it's not as much like a test as traditional tests are, but it still is going to require them to perform. So that's pretty much it. We have actually, some of our patients have become very into tracking their own medication. So they have a diary, or papers where they mark off, did they take their medication? And some of them are really, really like to have that and to be able to show you that, yes, they took their medication. We sometimes use stickies with smiley faces and things about the medication. So I would say we had very little problem with taking the medication.
You need also a urine sample, if I'm not mistaken.
We do need a urine sample.
Well hydrated.
Yes, as you know, that can be an issue sometimes for the patient that doesn't want to urinate in the clinic. which does happen to us occasionally. So we have used things where we send the family home with the urine container, and then they get the urine right before they come into clinic if it's a thing where the patient is refusing to go to the bathroom when they're in clinic. So yes, they will have to produce urine, and some don't like to pee in the cup. They think that's, you know, somehow scary. And so we have those big like bedpan things that they can pee in instead, or we put something in the toilet where that's like an artificial, you know, collection device that we can use. So we try to accommodate whatever is needed for the patient. I will say that a lot of the patients at our site, and we've had quite a few at this point, have gotten through most of these procedures without too much problem. The blood probably being the most difficult procedure, as everyone knows from long years of trials in Fragile X.
And Liz, you also mentioned the EEG and the eye tracking from the first study. Will there be any of that in this study?
Well, so Francie may be able to answer some of that. We wanted to add that on to the adolescent study. We are doing that at our site through a separate consent because we've been working with Lauren, and she thinks the data, Lauren Etheridge, who analyzes all of our EEG, and she thinks the data are really interesting and might be very supportive of a cognitive effect, but she needs more than 15 and 15 to be able to really do a good analysis. So we're collecting her more information in this study through a consent outside the study. We are not requiring it. Certainly if a patient has trouble doing it or doesn't like it, we don't have them do it. We only have the ones do it who kind of like to do it, and or who just aren't bothered by it. So more will be collected. I believe Cincinnati is going to collect EEG data as well. So that will be on a site-to-site basis.
Yeah, that's dead
Right
on.
now,
We're going to handle this as a sub-study within the larger study, much as Liz is doing already. And right now, having talked to Lauren, we're really looking at probably Cincinnati and Rush being the two sites that will be collecting these data sets.
so the folks who go to those other trial sites won't be doing that.
No.
Well, I think that helps to give people an idea of whether their kids can actually do the study.
Yeah.
The kinds of
Yeah,
questions
but you can go to Rush and not do the EEG and eye tracking are extra. They're just and so even if half of the patients in this study do that, we still have a huge amount more than we had from the phase 2 study. So the data will be really valuable. So we don't need, you know, every patient to do it, but having additional data is, you know, a good thing.
I think this study is generating a lot of excitement in the community, and we're certainly very excited about it as well. I think that a lot of people out there want to be part of this study, and we want to help them to be a part of this study.
Yeah, I think so far, I mean, you know, we just got to get all these other sites up and running so that we can start hitting our objectives. Obviously, the sites that are up and running are doing, you know, have pretty good lists of folks that we're running, you know, working through. And yeah, we'd like to not to have to fly everybody cross country to Chicago, but yeah, that is something else that we can mention. And, you know, again, as there is travel support, you know, within the trial and things like that. So if someone, you know, is hesitant to go somewhere, we are providing travel support and making sure that we'll do everything we can to get them enrolled.
Well, I just want to thank all of you for speaking with us today. It's a very exciting trial, and we're hoping to get as many people involved in it as possible. So thank you to Liz and to Chad and to Francie, and we'll look forward to talking more about this trial in the future.